Hey, it's my pleasure to introduce everyone. Niema Moshiri, assistant teaching professor in computer science and engineering at UCSD, and he's going to talk about computational methods in viral molecular epidemiology. So I'm going to turn it over. Thank you for coming. Oh yeah, there's stuff here. I should mention that you and I worked together before, and your research is in computational biology and specifically viral phylogenetics and epi, and that's where we work together. And now you're teaching a whole new generation of your tricks to a whole bunch of new generation of scientists and students. So really happy that you're doing that. And then I don't know if today you're going to talk about massive adaptive interactive text, but if you do, that could be pretty cool. So anyway, thank you so much for coming, Niema, and I look forward to the talk.
Yeah, thank you so much for the warm introduction. I appreciate it. Yeah, as Davey mentioned, we work together. I'm going to steal screen share if that's okay.
Yeah, sure. Go for it.
Perfect. There we go. And yeah, so for context, as Davey mentioned, my research, it's in viral molecular epidemiology, and specifically, I don't know as much about the low-level virology as folks like Davey, but my goal is, how do we make these public health or, virological type questions more computationally feasible? So basically building the toolkits, the methods needed to analyze these types of data sets in large scale. Oh, whoops, I just wiped out my, let me reshare in a second. I accidentally killed my presenter slides, my presenter comments. Sorry, one sec. It's almost as if I haven't been teaching remotely for a couple of years now.
No problem, Niema. Take your time. We have time. So
there we go. Okie dokes. And share screen. Perfect. Sorry about that. Cool. So yeah, so here's just a general outline of today's talk. Basically, I'm going to start by just giving a broad overview of what is viral molecular epidemiology. Then I'm going to talk about consensus sequence generation, which is one of the kind of first steps in modern viral molecular epi. Then I'm going to talk about, once you do this consensus sequence generation for a bunch of different samples and you've collected all these viral genomes, what do you actually do with them? Then I'm going to end by talking about kind of specifically my lab's goals and the relevant work. I didn't want to get too in the computational side of things for today's talk, just because I want to make sure things are accessible to a broad audience. But I'm happy to talk about the more nitty-gritty details if folks have questions about them afterwards. And I'm going to end by just kind of talking about what my perspective is for how to prepare for future epidemics and pandemics. Hopefully we don't have another pandemic in a while, but Prepare Institute, we want to be ready inevitably once something comes. So first, just an overview of viral molecular epidemiology in general. Here's an image of a phylogeny. So a phylogeny, just a fancy word for an evolutionary tree. It shows the evolutionary relationships between different species, where at the leaves of the evolutionary tree, you have the present day species, and then the internal nodes of the evolutionary tree represent ancestors, which typically we assume to be extinct. And this is a weighted graph. So the edges, those branch lengths actually have some meaning. And typically in a phylogeny, the branch lengths mean some unit of evolutionary distance. So that could be maybe years, or maybe more likely in this case, millions of years. It could be number of generations, it could be number of mutations, some metric where the bigger this metric, the more time has passed evolutionarily. So folks have probably seen some type of a tree like this in the past, dealing with species, but it turns out that we could use a similar framework when talking about viruses, where the leaves, instead of being present-day species, are samples that we've collected from patients, from wastewater, from wherever we collected those viral samples. And maybe the time span is much shorter, but it's the same exact concepts. So I'm going to shift gears slightly to talk about how we can think of epidemics from a computational framework, and then I'm going to tie these pieces together. So let's think of an epidemic, just kind of zooming out. Well, we can think before the epidemic happens, or just kind of simultaneously, just kind of the background is that there's some contact network. So there's some graph, some network, where nodes represent individuals, and edges represent all potential contacts across which a transmission event could potentially occur. And as you might imagine, these contact networks take very different shapes and forms depending on what pathogen you're talking about. So if we're talking about maybe HIV, if we're looking at sexual contact, maybe you would have a power law degree distribution where some individuals are highly connected, most individuals are very smallly connected. If we're dealing with some type of, something like COVID-19, maybe the graph is a lot more complete. So maybe if you're just in the same vicinity as people, you're connected. So maybe everybody is highly connected. Just the structure of this graph changes depending on what is the mode of transmission and what do those interactions look like. Okay, but assuming that we've modeled a contact network in some way, we can think of the epidemic itself as being this transmission network, which is a directed subgraph of the contact network. So it's a subgraph, so same nodes potentially, maybe a subset of the nodes and a subset of the edges. And it's directed in that we have some sense of who infected whom. So each of these edges is directional. And then we also have these labels on each edge because we have some temporal information as well. It's someone transmitted to someone at some time. So for many viruses, especially if we're talking about relatively short term, this network is going to look like a tree. And especially in the case of like HIV, we commonly assume that this transmission network is a tree. So no cycles in this network. But for long-term epidemics, or if we're talking about something like COVID, where there might be reintroduction of reinfection, it could potentially not be a tree. But either way, some subgraph where we have directionality of who infected whom at what time. Okay, so now let's tie back in the evolutionary tree. So if we zoom in on just a single transmission event, we can imagine that the host, the person who has the virus before the transmission event, there's some collection of viral lineages just kind of like evolving within that person. So we can imagine that the root of the evolutionary tree within this person is that initial virus that infected this person. And we can imagine that that spawned this kind of evolutionary trajectory within the host. And at present day, at the time of the infection, at the leaves, and I hope people can hopefully see my mouse, but I'm kind of drawing on I'm moving around my mouse to point things out. But at present day, at the time of the infection, we have a bunch of present day lineages within the host. And then at the time of infection, we can kind of imagine this bottleneck event where one of these present day lineages randomly gets plucked out and then infects the recipient. And then that starts a similar evolutionary process within the recipient, and the original host, the source, continues to evolve as well. So the virus continues to evolve in both individuals. Okay, so let's imagine that these individuals get sampled in some way. So maybe if it's HIV, maybe they come to a clinic and we take a blood sample that we sequence. If it's COVID-19, maybe we do a nasal swab, or maybe we sequence wastewater. Somehow we've collected a sample of the virus, and we can kind of imagine that this is equivalent to having a few random kind of lineages chosen from present time. And this is really the observed evolutionary tree. So even though there was truly this global evolutionary tree of all lineages evolving within all individuals, what we observe is a subtree. It's some subsampling of that full tree. And this is kind of the best we could ever hope to reconstruct, because these purple branches just never got observed. Okay, so evolutionary trees, we can think of evolution as going forward in time down the tree. And kind of putting it in a molecular framework, we can imagine there's some common ancestor genome that evolves over time. So maybe mutations are introduced along each of these branches potentially. And remember, with mutation, those mutations are heritable. So any branch of the phylogeny that they're introduced, it gets inherited all the way down. And by the way, feel free if folks have questions. I'm monitoring the chat. I've been teaching on Zoom for a while, so feel free to interject with questions if you want, and I can pause periodically as well. But yeah, so we have basically this generative process that we could theoretically describe as some random process in which each step is dependent on the previous steps. So you can imagine that I could generate a contact network under any number of network models. And then given a contact network, I can generate a transmission network under any number of epidemiological models, maybe an SIR model, maybe an SIS model, something more complex, where that model, kind of the spread of the pathogen, is conditioned upon whichever contact network that I had. And then similarly, as you saw, the evolutionary history of a virus is very tightly constrained by the transmission network. So I can kind of imagine that the phylogeny that I observed, it's generated according to some random process that's conditioned upon the specific transmission network that I observed. And then similarly, the viral sequences we can think of as some, so the present day viral sequences, it's some random process conditioned upon the ancestral viral sequence and conditioned upon the viral phylogeny. Oh, there was a question in the chat about a couple pages ago. So the question was, why are those purple subtrees observed? Did they die off or are they not important? Basically, when someone comes into a clinic and we sequence them, all we observe are just whatever we were able to grab. So these purple branches kind of, they represent, you know, if you sequence someone, or if you do a nasal swab or something, you're not collecting every single viral particle that was in that person. So these purple branches represent whatever viral particles that we didn't collect. So if I did a nasal swab, whatever stayed in their body. Yeah, great question. So yeah, so each of these steps is a random process conditioned upon the previous step. In reality, even though I would love to have all of this information about the contact network, the transmission network, the phylogeny, really the only thing that I can somewhat reasonably directly observe are the sequences, right? Even then, there's some error introduced when I'm trying to sequence a given viral genome, but I can somewhat confidently sequence the viral genomes from a given sample. And the goal of viral molecular epidemiology is kind of this backward step. So can I use these computational models that I've developed to try to infer properties of these earlier stages, hopefully in a way that gives me some actionable information? Okay, so maybe I'll pause there if there's just kind of like any pressing questions about anything I've talked about so far. This is kind of the framework that sets us up. Maybe I'll continue and I can always come back if people have questions at the end. Cool. So the first step, consensus sequence generation. How do I actually collect sequences of these viral samples? Well, first I want to introduce the concept of what's called a reference genome. So a reference genome, it's a high confidence assembled genome sequence that is constructed to be a representative example of an individual organism. And remember, all organisms of a given species, we have high levels of variability. So one person's genome is not identical to another person's genome. Just like that, one HIV sequence is not identical to another HIV sequence, but they should be almost identical. They should be very, very, very similar. And a reference genome is just a high confidence assembled genome sequence that's representative of the organism. It's not going to be exactly the same as any given sample you collect, but it should be almost identical. And, you know, reference genome assembly, it's been going on for the past couple of decades, with the first high confidence reference genome that's on a large scale being the human genome. And since then, we've assembled a bunch more genome sequences as time has progressed. And now novel genome assembly is not the most, it's not the most difficult problem. Like, people do it somewhat routinely nowadays. However, you'll notice that there's relatively big time jumps between each of these time points. And even in present day, even though it's much easier, there still is usually some kind of time delay. It takes some time to be able to sequence and assemble a high confidence reference genome of whatever you're looking at, up until the COVID-19 pandemic. So with COVID-19, we had something unprecedented in viral molecular epidemiology, which is that, you know, here I really want to highlight, this paper was published in February 3rd, 2020, you know, just a couple months after the very first, you know, unknown instances of this new pneumonia that's happening in Wuhan, China. So just months after we even knew that something was going on, a high confidence reference genome was assembled and published, which was extremely helpful during the pandemic, because this gives us a nice high confidence reference point across which we can kind of help out any future sequencing. So in the future, we expect that this will continue to be the case. If we ever have some novel pathogen pop up in the future, ideally we will sequence it rapidly, release a public reference genome for that novel pathogen, and then that'll help downstream analyses. But yeah, so February 2020, the SARS-CoV-2 reference genome was published. Now, how do we use this to help us in maybe San Diego, if I'm trying to sequence all the samples I collect in San Diego? Well, we can think of what's called a consensus sequence. It's not a de novo assembled genome. It's not a genome that I'm assembling just from the reads, but it's a sequence that I'm generating given a reference genome as context. So here in this figure, the little blue dashes represent reads that I've sequenced from some novel pathogen's genome. And the green line represents the reference genome. And instead of having to try to piece together these tiny fragments on their own, I can use the reference genome to help me reconstruct the consensus sequence, the genome sequence of this individual sample. And the reference genome is not going to be identical to this sample's genome sequence, but each of these reads should be almost identical. So what I could do is just map those reads against the reference genome. And then once I have those read mappings, I can just do a kind of sliding approach where I look at the first position of the reference genome and see in all of the reads that span that position, what was the most frequent letter that I observed? And then I output that as the first letter of this sample's genome sequence, the consensus sequence, right? It's consensus because I'm taking the most frequent letter. Then I move over to the next position of the reference genome and see what letters appear at in the read spanning this position of the reference genome. Whatever is the most frequent letter, let me output that as the next letter of my consensus sequence. And I keep going one by one until I reach the end of the reference genome, and I now have a consensus genome sequence that I hopefully am fairly confident. And throughout this process, it could be that maybe some parts of the genome, we just happen to not have any reads span it, or it could be that the reads don't really have a consensus. Maybe there's a high frequency of all the nucleotides. Maybe it's a contaminated sample. Maybe it's a mixed sample. So I can't really discern, is it A versus C versus G versus T? In this consensus sequence generation, I could use ambiguity. I can maybe output N instead of ACGT if I want to just designate there's some letter here, but I don't know what it is. And I could use other ambiguous nucleotides as well. But yeah, so basically I can scan across the reference genome and basically construct a consensus sequence from this given sample. And this method, it's been implemented in quite a few different ways, and kind of the gold standard tool that people are using nowadays is a tool called IVAR, created by Christian Anderson's lab over at Scripps Institute, Scripps Research. And yeah, basically this tool implements this strategy. And I'll talk more about how we can optimize these things a little bit later when I get to my lab's work. Okay, so up to this point, if you gave me one sample, I can run it through this pipeline and generate a consensus genome sequence. If you give me a bunch of samples, I could do the exact same process for all of them. So now I have all of these different genome sequences at my disposal. What do I do with them? First, to give a little bit of context, I want to emphasize that, you know, in the past with viral molecular epidemiology, we were dealing with the scale of maybe hundreds, maybe for a big study, maybe a couple thousand genome sequences. But something else that was kind of novel during the COVID pandemic, very directly supported by the fact that we published a reference, not we, but that a reference genome for SARS-CoV-2 was published early on, is that we had a kind of explosion of viral molecular epidemiology all around the world. So we had unprecedented levels of genome sequencing of the virus. So I just want to kind of put this 15 million genome sequences number here for context that this is the scale that we're dealing with. So let's talk about a potential kind of standard phylogenetics workflow. Let's imagine that I have a bunch of these unaligned genome sequences. Pretend that each of these short sequences, pretend it's actually a complete viral genome. They're very similar, but they might have some substitutions, some insertions, some deletions. The first step is typically multiple sequence alignment. I want to detect sequence homology by performing multiple sequence alignment, which kind of lines up the positions relatively nicely by putting these gap characters in the middle. And then after multiple sequence alignment, you typically do what's called phylogenetic inference. So given the multiple sequence alignment, how do I infer phylogeny that hopefully best describes how these sequences came about, best describes the evolutionary history? And this phylogeny that I get, it's an unrooted tree. Ideally, it's an unrooted, perfectly bifurcating tree. Hopefully, it's a perfectly binary tree. But in practice, there could be polytomies, there could be all sorts of mess. But regardless, it's some unrooted tree that I get that describes the evolutionary history. And then ideally, I want to be able to detect what was the most likely common ancestor. So then there's this non-trivial step of, given the unrooted phylogeny, how do I determine which point in this tree is the optimal root? And that kind of defines the directionality of the evolution. And in addition to that, there's all these other downstream analyses. So maybe I want to perform transmission clustering based on the sequences. So this is similar to what we do with HIV, but we've also been doing it with SARS-CoV-2. Maybe I want to do transmission clustering on the phylogeny. So this is some stuff that I've actually worked with Davey in the past with, dealing with HIV, but we've also done it with SARS-CoV-2. So I want to do some type of downstream clustering of my individuals, see what populations maybe the pathogen is spreading the most rapidly in, all that good stuff. So in general, these two steps are the key bottlenecks, the multiple sequence alignment and the phylogenetic inference. Once you have these two pieces, everything else is at least somewhat reasonably scalable. But these two steps are kind of the key computational bottlenecks. And also just the sequence generation because of how many sequences we have. So for multiple sequence alignment, I want to kind of talk about this problem a bit. It was, remember, I'm sticking in these gap characters to kind of show sequence homology by lining up the homologous characters of every single sequence in one column. And not really relevant to this talk, but I just kind of take my word for it. There exists an optimal algorithm where if you gave me two sequences, each of them of length k, I could align those two sequences in big O of k squared. And big O of k squared, if you're not familiar with this terminology, this just means the algorithm scales quadratically. And I drew the square here because really what's happening is I have a k by k table that I have to fill up. So if it scales quadratically with the length of the sequences. It turns out that there's actually a similar algorithm, it's a slight extension of it, that if you gave me three sequences of length k, I could determine the optimal alignment in big O of k cubed. So it would be cubic with respect to the number of sequences. And kind of in short, it's basically I have a three-dimensional table. I have a cube that I'm filling in. That's k by k by k. And it turns out I could keep doing this generalization. And if I wanted to optimally align n sequences of length k, where n is the number of sequences, k is the length of the sequences, this exact approach would be roughly big O of k to the n. So it would scale roughly k to the power of n number of operations that I would have to perform. So that seems reasonable, right? Problem solved. I have a solution. Unfortunately, not so much. So remember with COVID-19, if I'm looking at the SARS-CoV-2 genome, I have roughly 29,000 letters long of a genome, and the number of genome sequences is over 15 million. So if I wanted to, do 29,000 to the power of 15 million calculations, even assuming that each of these operations is 1 nanosecond, that's longer than the existence of the universe, a little bit too slow for real-time public health intervention. And for context, there also do exist heuristics, so algorithms that perform multiple sequence alignment that are not guaranteed to give you the optimal alignment, but hopefully give you something good enough. And these heuristics typically scale roughly quadratically with the number of sequences. But still, when we're getting to that 15 million point, that's getting pretty big. And, I'm hoping in the future, we'll just continue sequencing, ideally, every single sample that we collect. So we want this n term to get really big from a public health perspective. So how do we build a system that's going to scale to that size? That's kind of a big, important question. And similarly, so phylogenetic inference, let's assume that I've somehow managed to create that multiple sequence alignment. Well, given a multiple sequence alignment, I want to infer a phylogenetic tree from it. And I can use models of sequence evolution to define the likelihood of trees. I can take a parsimonious approach. I can take a Bayesian inference approach. There's a few different ways that I could use mathematical models to kind of define some scoring function on different trees. But it turns out that determining an optimal phylogenetic tree from a given multiple sequence alignment, this problem is what's called NP hard. There's no efficient solution that exists yet. So to combat this, heuristics are typically used. So for Bayesian inference, what they typically try to do is do some type of an MCMC approach to estimate posterior probabilities of the trees. We have maximum likelihood inference, where you basically have some starting point and you make small modifications to it in order to maximize some likelihood score. There's maximum parsimony inference, where you define some parsimony score and you maximize that parsimony score. And these heuristics can still be somewhat slow. It seems like maximum parsimony is pretty promising. So there's a faculty at UCSD, Yatish Tarakia, who came up with this mat optimized method that is very, very fast. But even then, these data sets are kind of rapidly growing. So even then, it's still kind of this computational bottleneck. Okay, so I've given you all of that context of what are the types of analyses that we want to be able to do. And I've kind of highlighted what are the key bottlenecks. So now I just wanted to go over some of my lab's work, some of my lab's goals, and talk about how we've contributed to some of these different problems. So my lab's goals, broadly speaking, I want to be able to identify and characterize some of the key computational bottlenecks in viral molecular epi. So already we've highlighted a few of them that kind of popped up during the COVID-19 pandemic, but in the future, we want to continue identifying and fixing these key bottlenecks. And to fix them, one of my lab's goals is to develop novel algorithms and tools to analyze large-scale viral sequence data. And I want to make these tools and these pipelines as massively scalable as possible. I want to be able to solve this problem so that it doesn't matter if we have 1 billion sequences in the future. Hopefully, we can enable real-time surveillance just by having a super massively scalable system. So those are the overarching goals of my lab. And some of the work that we've done over the past couple of years to kind of fix some of the different bottlenecks in this pipeline, or how we've contributed to them. So for context, I'm just going to keep showing this generative process and the inference side, and I'll kind of show where I've contributed in various aspects of this. So first, the entire generative process. So if I wanted to generate synthetic data with which I could either benchmark existing tools or explore various hypotheses about interventions, whatever I want to do, let's imagine that I want to generate synthetic simulation-based epidemic data. So the entire pipeline, we built a framework called Favite that lets you simulate a contact network, a transmission network, a viral phylogeny, viral molecular data, sequencing data, imperfect sampling, kind of all of these real world features that you would want in a very flexible framework. So basically the framework itself doesn't make any model assumptions about any of the steps of the pipeline. It instead describes the pipeline in this abstract way, and each implementation of each component of that pipeline makes the model assumptions. So basically users are free to pick and choose which models and which model parameters are most appropriate for their specific study. And especially in the future, as novel models that better capture reality are developed, they can be integrated into this pipeline to enable even better, more realistic simulation experiments in the future. As you might be overwhelmed by this figure, Favite is not the most user-friendly tool, and it's very, because of its flexibility, it's kind of hard to design experiments that use it. So more recently, with some of the students in my lab, we've been developing this framework called Favite Lite, which is a less flexible version of Favite, but that less flexibility, we enable a little bit more guidance based on some of the common features that we typically see users actually utilize in practice. And by reducing some of the flexibility, we've made it orders of magnitude faster and more memory efficient. So we're still currently finalizing the Favite Lite pipeline. We used instances of it in some of the past work studying COVID origins, and also right now studying HIV in San Diego. But it's still kind of being developed, and a manuscript is in progress. Now, kind of zooming in on each of those individual components, what if I wanted to just simulate contact networks? So how do I actually sample contact networks under various network models? It turns out that some of the built-in, people on this call might have used Network X or similar tools to simulate networks. It turns out that these tools, they're great for small scale epidemics, but if I want to do something that's like country scale or global scale, they blow up in memory and in runtime. So I developed a toolkit called Niema GraphGen, for lack of a better name. I was just kind of in a rush to push it out. That it implements many standard network models, and it lets you simulate contact networks under many of these different models, but much more memory efficient and much faster than existing toolkits. So in terms of memory efficiency, it's typically orders of magnitude better than many of the existing tools. And in terms of runtime, it's orders of magnitude faster than Network X, which is kind of the most standard one that people use. And it is a little bit faster than iGraph, which is another less common, but more efficient framework that exists. Then for the transmission network step, imagine you're given a contact network and you want to simulate an epidemic along that given contact network. Students in my lab are developing a tool. So I developed a tool called Niema favorites that is basically like a wrapper around an existing simulation tool that makes it more user-friendly to design an epidemic simulation. And then students in my lab are designing a web app that's going to then integrate into this Niema favorites framework and let users kind of in an interactive graphical user interface design their own epidemic. So, designing the states of the model, designing the transition rates, all those more kind of epidemic specific properties, they can then generate much more complex models and then simulate according to those models. Then given a transmission network, how do I simulate a phylogeny? The existing tool that was doing this before, the standard tool was one called Virus Tree Simulator, which worked well for small epidemics, but it ran into some major scalability issues. So I developed a tool called Coatran that's still kind of in active development, but a preprint has been out for a while. That's orders of magnitude faster than the existing tool. And that's kind of all that I've done in the generative side of things. But then in the inference side of things, now how do I actually analyze real data sets? I'll first maybe talk about the viral sequences. So how do I even generate viral sequences? As I mentioned earlier, the first step of any of these consensus sequence generation pipelines is mapping reads to a reference genome. So I've collaborated with folks that work on hardware acceleration in the computer science department to come up with various hardware accelerated solutions for DNA sequence pattern matching that we've integrated either for perfect matching as well as for sequence alignment. So here I'm showing kind of the key figure from an FPGA-based accelerated sequence trimming tool. So trimming is typically the first step of any sequence analysis, where this tool that we've developed, this FPGA-based tool, has orders of magnitude higher throughput than existing trimming methods. So FastP is typically considered one of the more efficient CPU-based read trimmers, and we have orders of magnitude faster trimming compared to that gold standard. And then now that you have a bunch of trimmed reads, what if you want to do de novo genome assembly? So this is not consensus sequence assembly. This is de novo just from the reads. How do I do genome assembly? So with those same collaborators, we built a de novo genome assembly acceleration method that uses near memory computing. And compared to the standard, so here we compare hardware accelerated speedup with respect to MegaHit, which is an ultra-fast CPU genome assembler for various steps of the genome assembly algorithm. And as you can see, our solution, which we've shown in, I think so each of these different colors, we get, so each color is a different instantiation of this implementation. The different numbers of specs is just details in the implementation that you can kind of vary. And we get, between, 1.5x to roughly 8x acceleration compared to what is typically considered to be the fastest CPU-based genome assembler. Then with regard to pattern matching for sequence mapping, so if I have a bunch of reads and I want to map them to a reference genome, for exact pattern matching, we were able to accelerate it using what's called hyperdimensional computing. And compared to many standard tools, we were able to get orders of magnitude speed up. Kind of for context, Minimap2 is typically considered to be one of the fastest CPU-based read mappers. And this hyperdimensional computing implementation that we've implemented on GPU, FPGA, and ASIC is able to do pattern matching orders of magnitude faster. And similarly, if we want to do specifically short read alignment that implements, so this previous one, it only supported exact pattern matching, but if you want to generalize it to sequence alignment using Smith-Waterman alignment, we have this salient FPGA solution that doesn't just do exact pattern matching, but also incorporates Smith-Waterman sequence alignment. And again, when we compare it to some of the gold standard kind of most used read mappers, we get orders of magnitude higher throughput. So it's orders of magnitude faster than what you're doing on CPU. And then RapidX is yet another kind of contribution we've made on the read mapping front. It's an in-memory ReRAM processing accelerator for sequence alignment. And kind of same story, we compared it to existing read mappers, and we get orders of magnitude higher throughput than what's been seen before. RapidX, we not only compared it with short read mapping, which you see in the top, but we also compared it with long read mapping. And even though the speed up's not as significant as with short read mapping, it still is consistently, with the exception of this one data set, it's typically faster than all existing tools. So those are all hardware acceleration solutions that we've developed that still are kind of more on the research side of things, but we're hoping to integrate it into real world pipelines to kind of make it easier for someone to take it from bench to analysis. So still, it's a little bit too non-user friendly to deploy, but that's kind of the next steps of those projects. But what about tools that already exist? Already a bunch of CPU tools exist. Each of them has their various trade-offs in terms of runtime and accuracy. And setting up all these different tools, it's very hard for a public health official or for a biologist to necessarily have to manually set up all these things on their own server, get them running. I'm sure all of us have had that headache at some point. So another tool that we developed is called by Reflow, which is basically an Amazon Web Service-based pipeline that the user can pick and choose what read mapper they want to use, what read trimmer they want to use, what approach they want to use for variant calling or consensus sequence calling. And you basically pick and choose which tool you want to use for each step of the pipeline. And by Reflow, when you give it all of your samples, will automatically determine how many AWS EC2 compute resources it needs to allocate. And it kind of dynamically scales the EC2 allocations. EC2 is kind of, for folks that are not familiar, it's the AWS kind of compute instance system. And what by Reflow does is it scales how much computational resource is allocated for your job, so that if you double the input size, you hopefully don't double the runtime, you're just doubling how many resources you allocate. So here, in the left plot, I want to emphasize that the horizontal axis is log scale, but the vertical axis is linear scale. So you have close to constant runtime, the overhead being just by Reflow handling an order of magnitude more inputs that it has to kind of send off to wherever it has to go. And as you can see, the total cost in US dollars scales linearly. So basically, it's showing that, yes, you give it 10 times more data, it'll scale the compute resources to analyze 10 times as much data in roughly the same amount of time. So you don't have to worry about kind of configuring your own environment. So that's just taking prepackaged tools, running them in a more kind of user-friendly and elastic manner. But what about actually speeding up the analyses? So for the viral consensus sequence generation pipeline, it turns out that a lot of the tools that are being used in the standard pipelines for this are kind of built on tools that were originally created for human genomics. And human genomics is a much different problem because the human genome is orders of magnitude longer than even the longest virus. But because of how short a viral genome is, we could actually do viral genome consensus sequence calling kind of on the fly. So I saw on the attendance list, there's a lot of students that are taking my class that I'm teaching right now. It turns out that you could basically just allocate all of the memory you need in one array up front. And as you stream in the reads, you could just kind of increment the counts of each of the bases on the fly. So I implemented this idea into a tool called Viral Consensus, and the preprint is out on bioRxiv, currently under review. And in terms of runtime, we get orders of magnitude speed up with respect to what's considered typically the kind of gold standard pipeline. And importantly, it has constant memory overhead with respect to the data set size. So it doesn't matter if you double or even multiply your sequencing depth by 10, sure, your runtime is going to take a little bit longer because you have to analyze more sequences. But the memory complexity stays constant. The memory only varies based on the length of the reference genome, which for the same virus will not be changing. And you'll notice that I'm able to analyze, 10 to the 6 reads in roughly 1 second, 1 to 2 seconds. So yeah, much faster than kind of the state-of-the-art tools. Now, how do you go from viral sequences to a viral phylogeny? Well, the first step that you have to do, as I mentioned before, is multiple sequence alignment. So prior to the COVID-19 pandemic, people typically used multiple sequence alignment tools that don't really make assumptions about the sequences that you're giving them. So those tools were built to be able to align any arbitrary sequences, no matter how distantly related they might seem. For viruses, however, if I have a bunch of SARS-CoV-2 genomes, these are almost identical. So I don't necessarily need a solution that is generalizable to completely arbitrary random sequences. I can take advantage of the fact that all of these sequences are almost identical. So I built this tool called Viral MSA that takes a reference-guided approach. It basically wraps around existing long-read mappers to map each of the genome sequences that you have against the reference genome, and then kind of uses the reference genome as anchors to reconstruct a multiple sequence alignment. And Viral MSA, it's able to keep matches, mismatches, and deletions with respect to the reference genome. It omits insertions with respect to the reference genome, but if you anticipate that you're going to have a lot of informative insertions, you can always adjust the reference genome accordingly. So on the left, you see that it's orders of magnitude faster than any of the existing tools that have kind of been used in this space. And on the right side, you can see that, so the Mantle test for accuracy, this is basically determining what is the Spearman correlation between a distance matrix computed from the true multiple sequence alignment versus the one that I inferred using either of these tools. And you'll notice that the accuracy of the multiple sequence alignment itself is negligibly different compared to what's typically considered the best practice tool. And actually not shown in this figure, but when we then infer phylogenies from the multiple sequence alignments, the phylogenies inferred from the Viral MSA multiple sequence alignment had slightly higher accuracy than the phylogenies inferred from the mapped alignment. But yeah, orders of magnitude faster, seemingly negligible difference in accuracy. Then for viral phylogenetics, how do I actually speed up some of the tools that deal with viral phylogenies? My lab built this toolkit called TreeSwift that is a Python package for doing highly scalable, very memory efficient and time efficient operations on a given phylogeny. So if you have an evolutionary tree, you can use TreeSwift to implement various algorithms that you might be interested in on this phylogeny extremely quickly. So compared to the existing tools, it has more functionality, and it's typically, it's consistently the fastest and most memory efficient tool out of the bunch. And then lastly, okay, so the transmission network, how do I infer a transmission network from a phylogeny? Well, it's hard to determine what the actual transmission network is, but as a proxy, molecular epidemiologists usually do what's called transmission clustering, where usually you try to cluster individuals based on similarities in their viral genome sequences. And HIV trace, which is kind of the standard tool for doing this with HIV, the same technique has been kind of adopted to other pathogens as well. It starts by taking a multiple sequence alignment, computing all pairwise distances under the Tamura-Ney 93 model or the TN93 model of molecular evolution, and then linking individuals if their TN93 distance is below some threshold. So the bottleneck here is computing all pairwise TN93 distances. Once you have those, it's very easy and very fast to actually infer the transmission clusters, but computing those distances is very slow. So collaborating with the same hardware acceleration lab, we developed an FPGA accelerator that, given a bunch of sequences in a multiple sequence alignment, is able to compute these TN93 distances orders of magnitude faster and much more energy efficient than existing CPU-based tools. And then what if you have a phylogeny? Can you then perform transmission clustering on the phylogeny itself? So to answer that question, we developed a tool called TreeCluster, which, given a phylogeny, implements various clustering algorithms on the phylogeny. So it basically takes the clustering problem and defines it as a series of different tree cutting problems. And so we actually have counterparts to HIV traces clustering method. There's other tools such as Phylopart and Cluster Picker that we also have counterparts of them implemented within TreeCluster. But as you can see, it's orders of magnitude faster than the existing tools as the dataset size grows. And importantly, the growth rate of tree clusters runtime is slower than the growth rate of the other tools, meaning this gap is going to continue to widen as the dataset size gets bigger. So that was just kind of me throwing a bunch of different things that we've been working on. Hopefully some of it stuck, hopefully some of it you found interesting. But kind of what I think is maybe most important to this talk is how do we prepare for future epidemics or pandemics? And I think depending on who's speaking, they're going to have different perspectives. I'm coming from the computational side of things. So clearly my recommendations are going to be computationally focused. But yeah, from my perspective, I think the biggest thing is we need to develop an open source database system for all viral sequence data. So right now, the whole world is using a platform called GISAID, which is a really good, nice open system, but it has its flaws and it's not open. I can't contribute to it. I can't really modify anything in it. And you have to kind of request access to it. It's not an open science type framework. So I think we really need an open framework that anyone can access the database. Maybe if you have a sensitive application, maybe the CDC or the NIH or whatever can have their own private deployments, but interoperable so that you can either maybe share data between labs or somehow just make it a lot more open than the system currently is. And really what I think is instead of just storing genome sequences, which is what GISAID does, we need to store all mapped reads. So a lot of my research has worked with COVID origins, and there's a lot of issues where some of those early sequences, prior to there being like a lot of kind of standard infrastructure for this type of stuff, the problem was they only store the genome sequence. So if they made any bioinformatics mistakes, maybe there wasn't really enough sequence coverage at any of the given positions of the genome to make a call, but they did it anyways. That signal is completely lost. But because all of these genomes are almost identical to the reference genome, you don't necessarily have to store the raw sequence data. You could theoretically just store the cigar strings, which are basically deltas with respect to the reference genome, as well as insertions. So ideally, I think my big picture vision would be to have users run Minimap2 locally to map reads, or maybe even just have like a upload SAM file button, or like, upload FASTQ file button, and have a WebAssembly applet kind of map reads on the fly without the user even realizing. And basically, as fast as you can even send the data to get uploaded, it'll do the read mapping on the fly. And then that way we'll have kind of long-term storage of the raw data rather than just those final outputs. Ideally, the database should perform trimming and variant consensus sequence calling on the fly. So as I mentioned, I implemented viral consensus. This is super fast. So you could theoretically do these things extremely quickly in a viral database. And I really want to continue accelerating, both in terms of algorithms and in terms of hardware, various bioinformatics tools that are of importance. As I mentioned kind of at the beginning of my talk, I do a lot of work kind of developing simulation toolkits. I think so far I'm not as knowledgeable about how we can actually take the results of those simulation experiments and make them actionable public health policy. So I think integrating the theory and the actual practice, finding better ways to do those in corporations, I think that's going to be especially of utmost importance. So maybe I'll go ahead and end it here since we have like 10 minutes left. But yeah, thanks everyone for listening to me ramble about algorithms for 50 minutes. And yeah, hopefully I'll answer any questions that folks have.
That's great. Thank you so much, Niema. That was a great talk. So yeah, questions are pretty much open forum. So if anyone has any questions, they can just, speak out loud, or I know there was one recently put in the chat. So maybe we'll start with that one. So
Yeah.
Argus had asked, is it that much more burdensome to store the entire BAM?
Yeah. So BAM files, if you store all of the actual sequence data from the reads, it would actually start blowing up in memory, like in storage space. But SAMtools natively has what's called the CRAM format, which is doing exactly what I'm describing over here, where the CRAM format is a more compact equivalent, where given that you need a reference genome to decompress it, it basically does a reference guided storage and compression. So anything that matches the reference genome, it doesn't store in the output file. And as a result, you can, I don't have the figure here. I have another project, maybe I'll kind of just annotate on the screen, where I built a prototype of this idea where it'll kind of take in either a FASTQ file that'll do the read mapping on the fly, or it'll take in a BAM file or a SAM file, and it'll convert it to a CRAN file and store it in a SQL database. And I think the result was like maybe a few hundred kilobytes per something like 100x coverage. I forgot if it was 10x or 100x that I tested. But yeah, it becomes actually quite small, like the size of a song, like an MP3. You could store all of the reads of a given viral sample. Yeah, thanks, Gene. Thanks for coming.
And then Niema, I don't know if you saw that too. Gene also put in a site here that has an NIH funded, bv-brc.org.
Oh, that's great to know. I'm gonna definitely take note of that. Maybe for the rest of the audience, I'll just post it here as well. I haven't looked into this, but I'll definitely take a look at it. That's super helpful.
It has all the virus sequences, bacteria, and others that are present in GenBank. So something to be aware of for sure.
Yeah, yeah. And to Argus's point, so awesome. Similar to bitmapping a dictionary, except your dictionary is reference. Absolutely. It's the exact same concept where, if I have the reference genome, I don't have to store any of the matches to the reference genome. Unfortunately, with viruses, it's not like human genetics where we have like these large scale rearrangements and we have, I don't know, duplication, insertion, STRs. With the virus, it's almost all identical. So it actually compacts really, really, really nicely. A great question. Any other questions from folks in the audience?
This is Davey. I just have a quick question. What would it, if we wanted to build your wish list database at UCSD, what would that sort of look like in terms of resources? And maybe you don't need to answer that now, but maybe you should think about it and then we could talk.
Yeah. So I think actually the hardest part is buy-in of expertise. So my problem is I'm coming primarily from like an algorithms and systems programming background, and I don't actually have that much database background. But I think, and the problem is for the database faculty, I've actually pinged some faculty about this in the past. It's kind of too trivial of a database problem for database researchers. So I think if you have the matching of expertise, if you get a database person that's excited to contribute to this, I think it's actually quite easy. So I mentioned that ViruDB prototype that I built. This took me maybe like 2 weeks to build, and you could deploy it on, not even too beefy of a server. The vision that I have an undergrad working on some other projects, compiling some tools into WebAssembly web apps. Basically, the end goal would be the server would just sit. It doesn't have to actually do too much computation on its own. The WebAssembly applet on the user side would do the heavy computation. So you're actually going to be kind of using the user's own web browser to be your compute node, which per analysis is maybe weaker, but every single user that uses it is basically doing it on their own. So it actually, you can do it like very cheaply and with very little maintenance.
Okay, cool.
Yeah.
Any other questions?
Well, thanks, Niema. This has been great.
Yeah, thank you for having me.
Super good work. Yep. Good to see you. Thanks, everybody. Have a good weekend.
I'm just going to quickly introduce the next seminar. Yeah, no problem, Davey. So we have a seminar next month, March 24th. Actually, next month is Wednesday already. But on March 24th from 1 to 2, we'll have Dr. Leslie Lewis here, who's from the newly founded Homelessness Hub, and she'll be speaking about public health impacts in homelessness. So I hope you all can join us then. Thank you all for coming today. Have a wonderful weekend, and we really appreciate you all being here. And Niema, great talk. Thank you very much.